Hi, my name is Brody Quinn,
I'm Alexis Hand.
I'm Melissa King.
And I am Holden Wood.
and this is our presentation on the comparison of microbiome diversity of Canis latrans, also known as coyotes, fecal matter in Monterey County. Coyote populations have been under threat by issues from urbanization and the lack of their normal diets that they function off of. Anthropogenic, also known as human polluting foods, are causing severe microbiome influences in large metropolitan cities, as seen in Scott Sugden's scientific journal titled "An Altered Microbiome in Urban Coyotes Mediates Relationships Between Anthropogenic Diet and Poor Health." The data was collected from two different locations in Alberta, Canada, that contained urban housing versus rural outskirts of the city to analyze the changes in the coyotes. The urban areas saw more severe cases of more malnourished coyotes that have indicators within their bacterial composition after they were dissected and samples were collected. Additionally, an article by Michelle J. Russell titled "The Prevalence and Characterization of Escherichia E. coli and Salmonella strains isolated from stray dog and coyote feces in a major leafy greens production region at the United States-Mexico border researched the impact of coyotes' feces on the agricultural industry. They discovered that although the strain of Shiga toxin producing E. coli that they were looking for, they did not find other pathogenic E. coli and Salmonella strains that were produced from the improper practices of mitigating microbial contamination on the crops. With Monterey County's steady population increase, the once rural outskirts will start to become developed and increase the interaction with wildlife and humans. Also, the heavy agriculture industry within the county will be threatened as less land is available for them to avoid interacting with the environment. According to our hypothesis, we predict that the Canis latrans fecal matter will provide evidence that species found in Monterey County are going to have healthier digestive tracts compared to those that are discovered scavenging around suburban, large human populated environments.
So to start our research, feces samples from coyotes were collected on January 22nd, 2020 in the trails on Engineer Canyon Road in Marina, California. The samples were added to a 3 milliliter LB broth and were incubated at 37 degrees Celsius for 24 hours. Unknown samples of coyote scat were available and categorized by appearance based on if there were bones, fur, twigs, or moss present. Using aseptic techniques, the unknown bacteria was isolated from the broth and streaked onto LB plates. The plates were then incubated at 37 degrees Celsius for another 24 hours. Aseptic techniques allows for a contamination-free environment for the bacteria to be isolated. Next, bacterial DNA was extracted by mixing a CFU, or a colony-forming unit, with 20 microliters of solution into a tube and placed in the thermocycler for 15 minutes. PureTac PCR-B tubes were prepared with 23 microliters of master mix, and which is made of 1 microliter of 27F primer, 1 microliter of 1492R primer, and 2 microliters of water. And then 2 microliters of microlysis in a CFU, and once again placed into the thermocycler. Standard procedures were followed to prepare the 10% agarose. Then 5 microliters of sample was mixed with 3 microliters of dye, loaded and ran at 120 volts for about 35 minutes. DNA was isolated by using the PCR reaction cleanup method, using the PureLink PCR purification kit. The DNA was sent and sequenced at the Molecular Cloning Laboratory in San Francisco. The sequence was inserted into the NCBI database using the BLAST, specifically the nucleotide BLAST, to compare and identify the bacterial DNA. Then the nitrate reduction test was executed by inoculating a broth that had a Durham tube and incubated at 27 degrees Celsius for 48 hours. If bubbles were present, the nitrate was reduced to nitrogen gas. If not, then drops of reagent A and B were added. If no change after 10 minutes, then a pinch of zinc powder was added. If the broth turned red after the addition of reagents A and B, then there is a reduction to nitrite. If the broth turned red after the addition of zinc, then there is no nitrate reduction. To perform the anaerobic test, standard aseptic techniques for streaking and inoculating broths were performed. One streaked nutrient agar plate and one inoculated LB broth were placed into aerobic conditions to grow at 37 degrees Celsius for 24 hours. A different streaked nutrient agar plate and inoculated LB broth were placed in the gas pack E2 anaerobic container system to grow under anaerobic conditions. Next, the Kirby-Bauer was performed by comparing the broth to the McFarland standard, then diluting the broth containing the bacteria with saline until an absorbance reading of 0.239 absorbance was obtained using the spectrophotometer set at 600 nanometers. 100 microliters of diluted broth was pipetted onto the plate and spread around with a hockey stick and turntable. 8 antibiotics, including streptomycin, amoxicillin, gentamicin, penicillin, chloramphenicol, tetracycline, neomycin, and erythromycin were placed onto the plate, then incubated at 37 degrees for 48 hours. The zone of inhibition accounts for the bacterial abilities of resistance to the antibiotic and were calculated and recorded after incubation. After all this, we took the data collected and input into Excel to create the graphs for our project.
This graphic shows the frequency of different genus of bacteria observed in the fecal samples. After viewing our sample data, the majority of bacteria seen in the sample was gram-negative Escherichia, with a percentage of 47% abundance in the fecal samples. Second was a gram-negative Acinetobacter with a percentage of 29% abundance. The final bacteria that was seen in the fecal samples that was worrisome was gram-positive Bacillus bacteria that was about 12% of the proportion. This figure shows the results of antibiotic resistance. We can see that the different antibiotics and their zones of inhibition for bacteria in fecal samples. This figure gives us insight on what food coyotes are consuming and how it's seen through the fecal samples. Most foods coyotes eat are creating a resistance to basic types of antibiotics. This occurs from the foods that we have benefits of resisting those bacteria. In our graphic provided, we can see that all of the bacteria are resistant to antibiotic penicillin, showing a 0 millimeter zone of inhibition. The results of the nitrate reduction test reveal 8 out of 8 of the bacteria tested had no nitrate reduction abilities, as well as 8 of the 8 bacteria that were able to reduce nitrate to nitrite, and 2 out of the 8 of the bacteria had the ability to reduce nitrate to nitrogen gas. The abilities of the bacteria to perform denitrification can be an indicator to the overall diet and the health of the coyotes. It has been found that high amounts of nitrate and isotopes of nitrogen reveals that a main source of nitrogen is coming from inorganic fertilizers. Due to the various surroundings of agricultural fields, this can be a major source of nitrogen finding its way into the digestive system of the coyotes and salinas. Therefore, it would be beneficial for the coyotes to carry bacteria that have nitrate reducing capabilities because most of the time nitrogen and oxygen are present together in the form of nitrate. If the nitrate is not reduced down to nitrogen gas, then it remains present in the environment, which can pose major problems for the groundwater.
Okay, so as my group member Melissa mentioned shortly ago, the most abundant genus present in Monterey County coyotes' gut microbiota are gram-negative Escherichia, gram-negative Acinetobacter, and gram-positive Bacillus. All of these bacteria can thrive under low nutrient anaerobic conditions that are consistent with the bacteria we'd expect to see present in healthy coyotes from rural populations similar to Monterey County, which I believe would be regions of Canada and Texas for our study. As seen here in the image on your left, for healthy coyotes, there's increased fusobacteria, increased anaerobiospirillum, and increased Sufrella, which are all phyla possessing anaerobic capabilities, just like the most abundant bacteria present in the guts of our local coyotes. The image also shows an increased protein in their diet with a decrease in anthropogenic food, being human scraps and leftovers, which is consistent with the Bacillus bacteria present in our sample, as Bacillus are typically found in decaying items like red meats and rotting roadkill. which indicates our coyotes are consuming less human scraps and protein diets on a regular basis. Although of the bacteria present in our fecal sample, the only bacterium of concern is Bacillus because they're typically always pathogenic, and too much of this particular bacteria present in the gut biome can cause coyotes and other organisms severe health issues. The fecal sample, when analyzed by Kirby-Bauer, indicates a zero zone of inhibition for the antibiotic penicillin. This is generally what we'd expect, as two of the most abundant genera present in our coyotes' gut biome were gram-negative. And penicillin works primarily against the thick peptidoglycan layer present in gram-positive bacteria. Our group and I then thought, were our Bacillus bacteria resistant to penicillin too? But upon reviewing the results, there were outliers present showing zones of inhibition for the antibiotic, which we'd say Bacillus are likely to be outliers showing less resistance to penicillin. Lastly, to provide a bit more evidence, Monterey County coyotes are healthy given their wide ranges of habitat. High amounts of nitrate and nitrate isotopes are found to be sourced from inorganic fertilizers common to our local agricultural fields. So we believe it's easy for these forms of nitrogen to find their way into the coyotes' digestive tracts. So it's beneficial for our coyotes to have nitrate reducing abilities, which the majority, 55% of the bacteria do, which supports our claim for healthy rural coyote populations in the Monterey County. And I believe Brody will take it from here.
Thank you very much. And to go back to our conclusion, we originally predicted that the Canis latrans fecal matter will provide evidence that species found in Monterey County were going to have healthier digestive tracts compared to those that were discovered scavenging around suburban and large human populated environments. The Canis latrans fecal matter provided sufficient evidence that Monterey County species have healthy digestive tracts compared to those that are discovered around urban environments. The most significant genus of bacteria, as Holden mentioned, was Bacillus, which was acquired through the consumption of nitrogen-rich food, such as dead animals or untreated plants, which is seen in salinas, containing infected soil organisms. For future work, samples would need to be taken from different parts of the coyote's digestive tract for accurate analysis on where the microbiome is playing a large role in keeping the body healthy. Many sources that looked into the evolution of coyote populations due to urbanization emphasize the importance of observing changes in the small intestine rather than the byproduct of the food broken down after it is excreted. Furthermore, there haven't been any sources based on the Monterey County that provide a basis to compare our data over time. So being able to keep track of this data on a 2 to 3 year collection can provide better graphs for change over time and benefit us in the long run. Lastly, using expensive tracking equipment could help keep track of the frequency of coyotes venturing into suburban areas around the coast of Monterey and see why they prefer to remain in these areas rather than returning to the more protected regions like Big Sur State Park. Here are references for our work. And lastly, we would like to acknowledge Jennifer Cato, Melanie Yu, Arlene Hoffa, Caitlin Tatsukawa, Rachel Durand, and Lou Zieberg for their efforts in providing our research for our group and with the help we needed to complete our work. Thank you, everyone.